The aim of this procedure is to generate and purify wild-type and mutant versions of the multi-subunit INO80 Chromatin Remodeling complex. This is accomplished by first isolating the nuclei from cells expressing flag epitope-tagged INO80 proteins. In the second step, nuclear extracts are prepared. And then, the wild-type or mutant human INO80 Chromatin Remodeling complexes are purified by anti-flag agarose chromatography. Ultimately, the subunit compositions of the purified INO80 complexes can be analyzed by Western blot or silver staining.
This method can help answer key questions about how the subunits of multi-protein chromatin remodeling complexes contribute to their overall activity.
This method is designed specifically for studying wild-type and mutant versions of the INO80 chromatin remodeling complex. But it can also be used to study other multi-subunit nuclear complexes, like the mediator.
The most critical step of the procedure is the nuclear extraction step. People usually run into problems because during the salt extraction step, people tend to add salt too quickly or too vigorously.
After culture in roller bottles, transfer the HEC-293 cells to a suitably sized graduated conical tube and spin them down for 10 minutes at 1000 times G and 4 degrees Celsius. After removing the supernatant, measure the volume of the packed cell pellet. Then, resuspend the pellet in 5 packed cell volumes of buffer A with gentle pipetting. And incubate the cells on ice for exactly 10 minutes. After spinning down the cells again,